everyone. So today we are going to discuss about the evidence of DNA as a hereditary material. Now, previously, when Mendel's work was published in the early 1890s, there was no evidence of there was no discovery of DNA. And once DNA was discovered, there was a question that whether DNA is the actual hereditary material in which the genes are present, from which causes to transfer genetic information from one generation to another generation or from one organism to another organism. So that was a huge question at that time. So evidence of DNA as a hereditary material was first confirmed by an experiment performed by a scientist named as Griffith. Griffith unexpectedly discovered, unexpectedly proved the evidence of DNA as a hereditary material. What he was actually doing was that he was utilizing his experimental material, which was a mice and the bacterium, which is known as Streptococcus pneumoniae. Streptococcus pneumoniae has two strands. Streptococcus pneumoniae has two major strands, which number one, Streptococcus pneumoniae has two major strands, which strains. Streptococcus pneumoniae has two major strains, which are known as the R strand and S strand. R strand and S strand. R strand and S strand. When it comes to the R strand, it is the type of Streptococcus pneumoniae. Strain basically means the variety or variety among organisms. Variety within the species is known as the strain. Let's suppose if you are having a strain of uh, what do we call it? Humans. There are different varieties of humans. So in that sense, there are different varieties of Streptococcus pneumoniae. In Streptococcus pneumoniae, we've got R strain and S strain. R strain is the one which is having rough appearance when viewed under microscope, and that rough appearance which is viewed under microscope is because of absence of polysaccharide coat. Due to the absence of polysaccharide coat, slime or capsule, the surface of Streptococcus pneumoniae of R strand appears rough when viewed under microscope, due to which this strain, which had a rough appearance, and that rough appearance was because of lack of polysaccharide coat, was known as the R strand. While when it comes to S strand, it is opposite of it, where polysaccharide coat is present in S strand, and that's polysaccharide coat present in S strand causes to give the S strand Streptococcus pneumoniae a smooth appearance, and that smooth appearance due to polysaccharide coat. Right. Now, when it comes to the R strand, when it comes to the R strand, it is a non-virulent strand. Non-virulent strand. Non-virulent strain, or we can also say this as non-virulent or non-pathogenic strain, and S strain on the other hand side is considered as a virulent strain, which causes infection, which causes uh, disease. So it is a pathogenic or virulent strain. Now, what actually happened in Griff's experiment was that he first injected what he actually did was he first injected the R strand, which was in alive form, into the mice. Due to which, what he noticed was that the mice remained alive because because the R strand was non-virulent. After which, when he injected S strand into the mice, which was a second experiment, where he noticed that the mice died because of the S strand injected. Now, in order to confirm whether the R strand, whether DNA, whether there is any sort of hereditary material which can be transferred from one strain to another strain, what he did was in the next experiment, he utilized a heat killed S strand. And when he utilized that heat killed S strand, what actually happened was that whenever the heat killed S strand was injected, the mouse remained alive, which was because the heat killed S strand was having the internal structure, the protoplasm of the Streptococcus pneumoniae of S strain killed, due to which the S strain was unable to perform its function and thus unable to cause infection in the mice, due to which the mice remain alive. The only thing which was left in the heat killed S strain was polysaccharide coat because it was non-living. On the other hand side, if we talk about heat killed S strain, in the fourth experiment, what he actually did was he utilized heat killed S strand and live R strand. Heat killed S strand, which was dead, and live R strand, which was alive. And when he uh, mixed both of these strands, the dead S strand and the alive R strand, what he noticed was that after a few moments, the mice died due to the heat killed assistant and live assistant which was injected in it. Now, in when this was observed, what he actually came to understand was that some sort of material, some sort of heredity material, genetic material must have been transferred from the dead S strand to the alive R strand, which had caused the mice to die because the R strain would have become virulent. R strain was non-virulent at first, and S strain is virulent. But S strain is dead, due to which that mechanism basically told us that there must be some sort of transfer of material from the dead S strain to the live virulent R strain, which caused the R strand, which was non-virulent previously, to become virulent as a result of ingestion of that material into the R strand. So this process of transformation was named by Griffith as the transforming principle. Transforming principle, or we also call this as transformation. Now, this was clarified that some sort of material was being transformed from R strand, from S strand to towards the R strand, which caused the R strand to become virulent. But the problem was over here that what was the nature of that heredity material, whether this um, heredity material was chemically DNA, was chemically RNA, or chemically protein. So that was something which was needed to be confirmed. Also, in order to confirm that, there were three scientists who came with the evidence of DNA as a heredity material. Name was Avery, McCallion, if I'm not wrong with the spell, and uh, Lennius. So these were some scientists. These were the three main scientists who actually performed an experiment in order to verify that whether that heredity material which was being transferred from in the transformation principle, in the transforming principle, was DNA or it was another sort of material such as protein or RNA. So when it comes to the experiment, what actually they did was they performed the same experiment. They performed the same experiment which was performed by Griffith. But what they actually did in that case was that they modified the culture. The culture in which the S strain was produced, the culture in which the R strain was formed, the culture in which the live R strain was formed, was having enzymes inside it, such enzymes which can either digest protein or digest RNA or digest DNA, due to which this could lead to disruption in any of their structure, and the transforming principle due to disruption of any of the structure might be stopped, as a result of no DNA or RNA or protein via this experiment. So this was a hypothesis given. Now what they actually did was, in the first experiment they utilized an enzyme which is known as protease. They utilize an enzyme protease, introduce the enzyme protease in the uh, culture in which the live they basically introduced the enzyme protease in which the S strain was present. So when they introduced the enzyme protease in the S strain which was present in the culture, they made this culture 99.9%, 99.98% free of protein, and the rest 0.02% was a protease on a chemical structure. So this was the work done by protease, that it basically digested the protein present inside the culture, right? And once the protein was digested inside the culture, the rest of the material which was formed inside it. So once we, you know, introduce protease inside it, then it basically caused to form a protein-free culture. Now, when the protein-free culture was injected into the mice, when a protein-free culture was injected into the mice, then that protein-free culture once injected into the mice basically caused the mice to die, as a result of which we conclude that transformation process was not hindered by the presence of protein, which means protein was not the hereditary material which caused the transfer of virulence from S strain to R strain. Now when it comes to second part, what we are going to have in the second experiment, what they did was that they utilized RNase enzyme. RNase is an enzyme which causes RNase enzyme causes to hydrolyze, causes to degrade the RNA. So when they introduce RNase inside the culture dish, they made the culture free of RNA, due to which when the culture free of RNA was injected into the mice, they noticed that still there was the same transformation process going on. There was no such big change. But when it comes to the third experiment, where they utilized DNase enzyme, they utilized DNase in order to hydrolyze the DNA molecule. So at that moment, what they actually observed was that they show they basically had seen a stop in the process of transformation which was occurring inside the culture dish. As there was a stop in the process occurring within the culture dish, the process of transformation inside the culture dish was stopped due to the introduction of the DNAs, as a result of which, when the culture was injected into the mice, what they actually saw was that the mice was alive when a culture free of DNA was injected into the mice, due to which no transformation process occurred and no transfer of hereditary material from the dead virulent S strain to the alive non-virulent R strain occurred, due to which it confirmed clearly that the DNA was a hereditary material which was being transferred from the DNA was a hereditary material which was being transferred from the dead virulent S strain to the alive non-virulent R strain. Now, this experiment was further confirmed by another follow-up experiment which was performed by Hershey and Chase. Hershey and Chase performed an experiment in which the material which they utilized was the E. coli bacterium, and alongside that What they actually utilized was a bacteriophage. What sort of bacteriophage was that? It was a coliphage. That coliphage possessed a single-stranded DNA as its viral genome. Single-stranded DNA as its viral genome. Single-stranded DNA as its viral genome. And that single-stranded DNA as a viral genome was found in the coliphage named as T2, because there are certain names of these coliphages. There are T2, T3, T4 bacteriophage, and we have double-stranded DNA bacteriophage as well, which is known as Phi 178. That is an exception. Mostly bacteriophages are single-stranded DNA, but Phi 178 is double-stranded DNA structure. So this is just an general knowledge portion. Apart from which, when it comes to the current experiment we are focusing on, over here, what they actually did was that they prepared a T2 bacteriophage culture. They prepared a T2 bacteriophage culture, and within that culture, what they actually did was they introduced a of radioisotope of sulfur 35, and in another culture they introduced a radioisotope of phosphorus 32. Phosphorus 32 in one culture and sulfur 35 in another culture, which was uh, which basically showed that which basically showed us that the sulfur 35, which was a radioisotope, would be becoming the part of the amino acids of the capsomere of the capsid of the bacteriophage in case of the first culture. And in second culture, the phosphorus 32, which was utilized, would become the part of the viral genomes DNA's phosphate group, due to which the DNA would become heavier and become a radioisotope. Now what they actually did was they prepared both of these cultures, which contain radioisotope sulfur 35 and radioisotope phosphorus 32. When they injected when they introduced an E. coli bacterium within that culture, they found that in the first culture, the sulfur labeled protein was sulfur labeled protein, which was having the amino acids having the sulfur 35, was left behind during the process of injection in the E. coli bacteria. As we have previously studied, that when the bacteriophage causes to inject its viral genome into the bacterium, the rest of the material which is left behind, the rest of the material which is left behind is rest of the material which is left behind is the capsid. It is basically the proteinaceous material which was left behind, and over here the material which goes inside is known as the viral genome. Now once the viral genome was goes inside the material, goes inside the cell, it causes the infection within the cell, and it does a number of processes, such that first it causes to hijack the DNA by destroying the DNA of the host cell by utilizing the enzyme endonuclease. That is not restriction endonuclease, but that is the endonuclease which causes to destroy the entire DNA. After which it integrate. After which, what it actually does is that it utilizes its own single-stranded DNA to produce its copies and produce all the structural components of the T2 bacteriophage. And once the structural components of the T2 bacteriophage are formed, and when they are assembled together in order to form a new match, a new virion, they basically what they actually saw was that the bacteriophage which was being secreted out from the E. coli bacterium were having the radioactive phosphorus 32, but that bacteriophage was not having the sulfur 35 which was present in the case of the E. coli bacterium. Of course, they compared both of these. So in first one there was no radioisotope, and in the second culture there was the radioisotope phosphorus 32, which was further which further basically proved us the point that the DNA was the hereditary material which was being actually transferred from the bacteriophage into the E. coli bacteria, and from that was being synthesized into newly formed virions which was being secreted, and that newly formed virion also contained the P32, which further proved this point. Now, when it comes to the evidence of this, this was done with the help of centrifugation machine. What they actually utilized was that they utilized a centrifugation process, a third centrifuge, in which the radioisotope being heavier tends to remain in the bottom, which is known as the pellet, and the non-radioisotope material was supposed to be in the supernatant. So what they actually did was when they uh, injected the first culture of bacteriophage which was having sulfur based protein into it, they saw that the E. coli bacterium did not have the sulfur containing bacterium, the sulfur containing proteins, the sulfur containing amino acids, or the radioactive isotope sulfur 35 in the pellet, due to which they couldn't find it within the pellet, thus due to which they were unable to, due to which they clarified that protein was not the material which was being transferred from the E. coli bacterium to the from the T2 bacteriophage to the E. coli bacterium to the new virion. But when it comes to the centrifuge machine, in case of second experiment, what actually happened over there was that in the pellet they saw the presence of P32. The presence of P32 was there, as a result of which the bacteriophage which was being transferred in P32, the bacteriophage viral genome was present in that culture, due to which the P32 being radioisotope was found in that culture, due to which it identified that DNA was the hereditary material which was being transferred from the bacteriophage into the E. coli bacterium into the new virion. So one more thing that I like to highlight over here, that the culture in which the S35 radioisotope containing bacteriophage was prepared was a different culture, and the culture in which the radioisotope containing bacteriophage was introduced in the E. coli bacterium was a different culture. Let's say the first culture was in which the bacteriophage were prepared. The bacteriophage were prepared in the first culture, in which there were the radioisotopes, and after that these bacteriophages were introduced into another culture which contained the E. coli bacteria. So this is how the experiment was performed. Alright, that's all about today's video. Hopefully we'll meet in another video.